Hello everyone, I hope you're having a wonderful day. So today's video is going to be on synaptic transmission and a little bit about neurotransmitters. So last chapter, or not chapter, but episode, we talked about an action potential and it traveling down a neuron. But now that's got to get to the end of the neuron and do something. So how does it send that signal from one neuron to the next? So the focus of today's episode is going to be going over synaptic transmission. And in doing so, we have to talk about neurotransmitters as well, because that's how this transmission process works. All right, let's hop right into it. So we're going to start out by just talking about the synapse here. So jumping right to the end of the axon. So if we're looking at this here, this would be the axon coming down here. This is referred to as the presynaptic neuron. Whereas down here then, this is the postsynaptic neuron. So this is the next one that we want the signal to travel along with. So here the signal is coming down, or the stimulus is coming down. Remember, we have degrees of myelination and all those. Here we see that myelination, and here it is synapsing on this next cell here. So remember the structures of neurons. These are called dendrites. Typically, presynaptic neurons synapse on dendrites. They can sometimes synapse onto the cell body. Sometimes they can even synapse onto the axon of the next cell. Depending on where they synapse, they get a different name. So, such as the one that synapses onto a dendrite is called an axodendritic axon or synapse. And remember, one neuron can have tens of thousands of synapses. So this is happening multiple times. So let's jump back into it now. So as this stimulus is coming down through here, remember it's an electrical current jumping from node to node, the action potential. Then we reach what's called the axon terminal. Up until this point, we had all sodium channels. So remember, sodium or potassium channels. One unique thing that changes down here at the synapse is these channels become calcium channels, or Ca+. 2+. So calcium channels. These are essential in releasing neurotransmitters. So inside the little presynaptic membrane here, we have these little vesicles. So these are called synaptic vesicles, and they're packaged with neurotransmitters inside. These neurotransmitters, let's say right now we're talking about acetylcholine. So acetylcholine is going to be packaged in this vesicle, and then if you remember the process of exocytosis, when the signal gets down here, these vesicles move down to the cell membrane, they fuse and release their contents into what's called the synaptic cleft. The neurotransmitter then binds to receptors on the postsynaptic neuron that then sends the signal in here. So here we are changing the polarization of the next cell. Let's say we're bringing in positive ions. Let's say we're bringing in sodium ions. That changes it and causes depolarization. And when you depolarize the cell, you can cause an action potential to occur in that postsynaptic neuron, which is the goal here. If you're sending a stimulus, you want to go neuron to neuron and send that signal to the next one. So a lot of things can happen here. You can have a lot of different neurotransmitters. You can have a lot of different receptors, and you can have different connections and so forth. But we're going to be talking about the general theme as we go down through. So why do we switch to calcium channels? Why do sodium channels become calcium channels at the synapse? That's the next part. So this all comes down to neurotransmitter release. So here is a great image of this process. So neurotransmitter is typically made, they can be made in the cell body, or they can be made by astrocytes that are surrounding these neurons. They're then packaged into these vesicles here. The vesicle transport brings them down through and stores them all in the presynaptic neuron. Down here, there's also a lot of mitochondria producing ATP, because this is a high energy area. So neurotransmitter is synthesized in the cell body or in the terminal down here. It depends. It varies from neurotransmitter to neurotransmitter. And then once it's down here, when it's made, it's packaged into a vesicle. Vesicles, are these cell membrane-like structures, and the neurotransmitter gets packaged in it. Remember, these Vesicles are a phospholipid bilayer as well. Next step is the calcium. So not shown in here are the calcium channels. I'll make this in green so it's easier to see. Calcium channels. When the action potential gets down through here, calcium rushes in here. Now, calcium is the signaling molecule which tells neurotransmitter to release. Sodium would not cause neurotransmitter release if sodium channels were present here. So these vesicles then move towards the cell membrane. As they move towards the cell membrane, there are these little snares. They're called V snares and T snares. So if this is a vesicle, they have these little proteins and they wrap around each other. And as they wrap tighter and tighter, think of them as like bread ties wrapping around each other. And eventually, fusion occurs, and the vesicle fuses with the cell membrane, releasing the neurotransmitter to the outside. So just the vesicle membrane becomes the presynaptic membrane. So you can see that highlighted right here. So neurotransmitter is then released. And then the vesicle can then come back and be repackaged and be remade and so forth. So it's a great little pathway that causes this. Oh, here down here you can see the calcium channel they made. So calcium is what causes vesicles to be released. It initiates the pathway for vesicle release. Now, we have neurotransmitters out in the synaptic cleft. Neurotransmitter can, one, bind to a transmembrane protein out here that has some ion come into the cell. That ion could be positive charge, it could be negative charge, or that binding could cause ions to come out. Those ions could be positive or negative charge. So neurotransmitters can be both excitatory and inhibitory depending on what happens at that postsynaptic membrane. When they bind to that protein or that ligand-gated channel, what does that protein send in and out of that next cell? So if we're sending positive ions in, we're depolarizing. If we're sending positive ions out or bringing negative ions in, we are hyperpolarizing and inhibiting that action potential from firing. And different neurotransmitters in different receptors do different things. So we have that neurotransmitter out here now. What happens to it? One thing that can happen, it can be reuptaken by the cell. So here is showing it here. So the neurotransmitter comes out, it diffuses out of that synaptic cleft, and then goes right back in and can be repackaged. So this is called reuptake. Another thing that can happen is degradation. So there's 1, 2 is then degradation. Degradation is, so let's use acetylcholine. Astrocytes, I believe, release acetylcholine esterase, which is the enzyme that breaks down acetylcholine and gets rid of it. And then the last one is diffusion. So remember, this is a space right here. That neurotransmitter can diffuse out, and once the concentrations are low enough, it's not going to send that signal anymore. So 3 different things can happen: reuptake, degradation, and diffusion. Now, a lot of stuff can modulate this process. Those are called neuromodulators. So a neuromodulator is something that changes something in this process. It could cause the synthesis of more neurotransmitter, cause the production of more receptors, cause a faster release. A lot of different things could happen to modulate this process. And those are not considered neurotransmitters, those are neuromodulators, because they modulate this whole process of, a big thing that happens in long-term potentiation is more calcium channels. So long-term memory is forming more calcium channels. You get more calcium coming in, you get more synapse, you get more neurotransmitter release, and more likely to send a signal in the next cell. So how do we send that signal in the next cell? And that's what we're going to talk about next. This is referred to as postsynaptic graded potentials. Remember, when we bind to the ligand or that receptor on the postsynaptic membrane, we could bring in positive ions, we can bring in negative ions, or we can send out positive ions or negative ions. So these postsynaptic graded potentials could be excitatory. So an EPSP is an excitatory postsynaptic potential, whereas IPSP is an inhibitory postsynaptic potential. So it all depends on whether you depolarize. I spelled that wrong. Or you hyperpolarize. So hyperpolarizing, you're inhibiting. Depolarizing, you're exciting. And now you can have multiple. So here is all the green ones are EPSPs, and they're all forming on the dendrites here, but then you have some IPSPs. And now you can look at the difference between temporal versus spatial summation. So it all depends on time and space. How fast do the inhibitory ones get there? How fast do the excitatory ones get there? And depending on the rate at which, whether it's the frequency or the time, it all comes down to whether you can fire an action potential in this next neuron. So temporal, this is time. Spatial is same time. So let's look at this. If we have two excitatory ones hit, at similar times here, so we have a bump, and let's say we're going away from this graph now. Let's say we then have another excitatory. Minus 55 is threshold here. We then fire that action potential. So temporal summation is, graded inputs over time. Now temporal, you could also have then that inhibitory here. And then you have another excitatory, another inhibitory, and then here it looks like two excitatories came in. So this would be spatial or same time. So this one came up double. So same time, two excitatory synapses come in, and boom, pops it up. You hit threshold. So all that matters is whether or not you hit threshold. Minus 55, minus 50 volts. You open those sodium channels, and then you fire that action potential. So that's IPSP versus EPSP. And then you can have different summations on different neurons. And then remember, there is a little delay. If we go back here, this is called synaptic delay. These neurotransmitters have to diffuse across the space. They have to bind to the receptors, and that receptor has to have an effect, whether it's depolarization or hyperpolarization. So that's why we have temporal and spatial summation. Some of these signals are received at the same time. Some are received at different times. Now, don't confuse this chemical summation with electrical. Electrical synapses do occur, and that's when you have a gap junction between two neurons. So you don't have the synaptic cleft where it needs to diffuse. So electrical synapses pretty much are instantaneous, like a normal action potential, whereas chemical synapses have a little synaptic delay to them. And this delay can be taken into account with temporal versus spatial summation of the action potentials. So next, I want to talk about different neurotransmitters that exist. So neurotransmitters are what are released by the vesicles. So neurotransmitters are broken down into their effects and their actions. I'm not going to go over a ton of neurotransmitters, but some just name off the top of my mind are acetylcholine, norepinephrine. You have, can't think of any right now, brain fart. So, but acetylcholine, noradrenaline, norepinephrine, glutamate, GABA, which is a butyric acid, endocannabinoids, there are lots of neurotransmitters. And I'm not going to go through each neurotransmitter now. I'm going to talk about overall effects. So main effects, 1 or 2. They can be excitatory, or they can be inhibitory. And this varies from neurotransmitter to neurotransmitter. Something like acetylcholine can have both excitatory and inhibitory function. So in our skeletal muscle, it's excitatory, but in our cardiac cells, it's inhibitory. It all depends on the receptor. So typically depends on the receptor for whether it's excitatory or inhibitory. What does that receptor do to the postsynaptic membrane? Does it cause depolarization or hyperpolarization? That's all that matters there. And then actions, they can be direct or they can be indirect. Direct means you're sending in ions directly. So whether they're positive or negative ions, the neurotransmitter binds to the receptor, brings in positive ions, depolarizes the cell, and sends the signal. Indirect is slow. So this one is faster. This one is slower. And this one's slower because it has to go through secondary messengers. So indirect, a great example of these are what are called G protein coupled receptors. Now, I don't have the time to go over G protein coupled receptors in today's video, but I might make a separate one sometime later, but it's not in the plans right now. What a rough idea what a G protein coupled receptor is. The neurotransmitter binds the receptor, then activates a G protein, activates an adenylate cyclase, turns on cyclic AMP, then cyclic AMP then could bring in calcium, could go to the nucleus and activate gene transcription, could cause a phosphorylation event. We're turning on enzymes, and all those effects could eventually lead to depolarization or hyperpolarization. But it's a pathway it has to go through, so it is much slower. But all these different things can happen in that synapse. So excitatory, inhibitory, direct and indirect effects, and all these things can summate, forming these graded potentials on those postsynaptic neurons. So this was a brief overview of synaptic transmission and overview of just the anatomy of a synapse. So I hope you got a good take-home message today. If you have any questions, just let me know. Thank you. Hello again. Thank you for watching my video. I'm glad you could come by. If you enjoyed this content, feel free to do that thing where you like and subscribe. Also, if you're interested, I prepare and record all of these videos live on Twitch. After the recordings, we do a post-recording question and answer section, and then sometimes I play games where any question is fair game as I game. Also, on Twitch, I'm a co-founder of a wonderful community called the Knowledge Fellowship, or TKF. Our goal is to seek and share knowledge with others while live streaming. We feature streamers who cover biology, law, physics, chemistry, geology, photography, health and fitness, psychology and mental health, rocket science and aeronautics, programming, and tons of different practical EDU streams. Feel free to check out TKF at the link in the description and join the very active Discord community. Hopefully, find something that interests you. With that, I'll see you all next time and have a wonderful day. And remember to seek knowledge and challenge everything.